Okay, hey Biochem. So this is day 3 of fatty acid oxidation, our final day, and we're going to be going over fatty acid nuances. So what happens if you don't have, or what happens if you do have double bonds, odd numbers, branch chain, and something that's called very long chain. You should also be able to compare fatty acid oxidation in the mitochondria versus a peroxisome, recognize the structures of ketone bodies, and then we're going to do 3 diseases today that are kind of spread throughout: ketoacidosis, MCAD, which is medium chain acetyl-CoA dehydrogenase deficiency, yikes, and X-linked adrenoleukodystrophy. Okay, so here's the outline. We're going to do MCAD first, and then we're going to do the fatty acid nuances, and then we'll do X-linked, or sometimes it's called ALD, or LALD, and then we'll end with ketoacidosis. Okay, so we're going to start with MCAD first because it kind of gives a review of beta-oxidation from last time. So over here is the pathway for beta-oxidation. And this disease, MCAD, which stands for, maybe I'll zoom in here, medium chain acetyl-CoA, so MCA dehydrogenase deficiency. Sometimes you might see this just as MCAD, leaving out the last D, just food for thought. Medium chain means that the acetyl-CoA that's going in is somewhere between 6 to 12 carbons. That's what medium chain is. So what this is affecting this enzyme right here. So acetyl-CoA dehydrogenase. That's this last part right here, or the middle part right there, the acetyl-CoA dehydrogenase. There's many different ones of these in your body, and they are specific for a chain length of the acetyl-CoA. So there's short chain, there's medium chain, there's long chain, and there's very long chain. So there's many different flavors of these, but in this deficiency, or in this disease, it's a recessive autosomal disease, I believe. Did I write that down somewhere? Yeah, so basically what happens is in your body, you have 2 genes for MCAD. That yellow is probably hard to see. You get one of them comes from your mom, and one of them comes from your dad. And as long as one of them is working properly, no mutations, then you won't experience this disease. But if you get a mutated enzyme from both mom and dad, then you'll have MCAD. Okay, so what it is, or what happens is you eat fat from your food, or you break down your body fat into these medium chain fatty acids. The first step in beta oxidation will begin the dehydrogenase of that, and then you can get energy from your cells. If you don't have MCAD, or if you have a malfunctioning one, you get little to no energy made from your cells. Why am I saying so much? So some signs and symptoms. Wow. We've got hypoglycemia, vomiting, sleepiness. Some of this can eventually lead to coma and even death. And it's made worse if the person who has this disease is fasting. Okay, so it has about a 25 to 60% mortality rate in children. and it's been linked to sudden infant death syndrome or other causes of just sudden death. If someone dies suddenly, particularly if they're a child, then they go back and they start looking for this particular deficiency. Okay, so how do you detect it and how would you treat it? So this is one of the diseases that is detected through newborn screening. And that's because of all the fatty acid oxidation diseases, MCAD is the most prevalent or most, let's say, most common genetic disease from fatty acid metabolism. And what they look for is low carnitine. Okay, so remember carnitine is part of the shuttle that gets the ACL-CoA group into the mitochondria. Well, if you have an MCAD deficiency, then you're probably not shuttling a whole bunch of carnitine over because there's going to be a buildup of fat in the mitochondrial matrix. Okay, so there's a buildup of ACL-CoAs, medium chain ACL-CoAs in the matrix because they can't be broken down. So you have low carnitine because there's no need to shuttle anything else in. And I think I've got treatment on this slide too. So low carnitine is what they're looking for in the newborn screening. There's a few different treatments, and some of them don't include any kind of drugs or prescription. Some of them are just strictly dietary. Not all treatments work for all. So I'm not a physician, obviously. So if you were interested in this particular disease and wanted to know more about it, obviously you need to dig a little bit deeper. But it relates to what we're talking about. So some treatment things are, if you have this, you have to avoid fasting. Or if you have a child with this, they cannot fast. They cannot go long periods of time without eating. They need to be constantly eating. And typically what they're going to be eating is a low-fat, high-carb diet. Some other extra treatments include carnitine supplements. And let's see. Yeah, I already talked about that it's, autosomal recessive disease, and it affects both boys and girls, but it can be managed if detected early. Obviously, with newborn screening, this disease can be detected, whereas before it was almost always would lead to death. Okay, happy thoughts. Moving on. All right, so last time we talked about beta-oxidation, where we only had a saturated ACL-CoA group going through beta-oxidation, but there's lots of different kinds of fatty acids. And so the first nuance, they're called nuances, is what happens if you have a double bond. So if you have a double bond in the ACL chain, it needs to be isomerized. If it's cis, it has to be isomerized to trans. Okay, and you'll notice, you don't need to memorize these enzymes here, but you'll notice this doesn't cost any energy at all. And the reason it has to be isomerized to trans is because it will enter in to the second step, which will only accept a trans double bond. So the substrate can only have a trans double bond. If the fatty acid ACL chain already has a trans double bond, then it just enters right in. One thing I do want you to notice is that for every double bond, I put DB for double bond, you're going to lose one FADH2. okay? Because when it gets to this round, that double bond is going to skip the first step and enter directly into the second step. So when you're doing your ATP math, every single double bond is going to cost you an FADH2. So for this acyl chain right here, it has 18 carbons. You're going to expect it will give you 9 acetyl-CoAs. I'm losing my train of thought. 8 rounds, so you would normally say 8 NADH and 8 FADH2, but you'll notice that there's a single double bond here, so we're going to, in that particular round, it's going to skip over the first step and enter into the second step. So you're going to lose 1 double bond for every FADH2. Okay, the next problem that can happen is if you have an odd chain fatty acid. So I'm going to draw an odd chain fatty acid out. Oh, let's do 13 carbons. So it's 1, 2, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, everything proceeds as normal. So we break off 2 carbons every round as acetyl-CoA. Okay, and then here's the last round. What I want you to see in the last round is we have 1, 2, 3, 4, 5 carbons. And again, we're going to break after 2. So what you have is this is going to be 1 acetyl-CoA, 2 acetyl-CoAs, 3, 4, and 5. But this has 3 carbons. And what that's going to lead to is instead of acetyl-CoA that has 3 carbons, it's going to give you something, or instead of acetyl-CoA that has 2 carbons, it's going to give you a CoA that has attached to it 3 carbons. And if you remember from organic chemistry, it goes methyl, ethyl, propyl, butyl. Propyl means 3 carbons, and so this is propenyl CoA. Okay, so everything proceeds as normal. Your number of rounds is essentially figured out the same way. The number of NADH is the same. The number of FADH2 is the same. The number of acetyl-CoAs will be dropped by 1, and you'll get basically the number of acetyl-CoAs equals the number of rounds for an odd chain fatty acid, and you'll always only get 1 propenyl-CoA. Okay, we have to talk about what happens to the propenyl-CoA. It's not lost energy. You'll notice that there are a number of carbon-hydrogen bonds sitting in there, so we don't want to waste that energy. It's going to go somewhere, but it's going to be a little bit of a process to get there. All right, so again, you don't have to memorize this whole series of steps, but what I do want to show you is that propenyl-CoA is going to be converted to succinyl-CoA. And the reason I'm showing all these steps is because it's going to cost you 1 ATP. So you're going to have to, for every propenyl-CoA, you're going to have to subtract 1 ATP to get to succinyl-CoA. Now I have the TCA cycle over here because this succinyl-CoA will enter in sideways into the succinyl-CoA, and we'll get a rotation from succinyl-CoA up to the top. So what you'll get out is 1 GTP, 1 FADH2, and 1 NADH. Did you get all that? So 1 propenyl-CoA is going to equal an NADH, just 1 FADH2, 1 GTP, and then it's also going to cost you an ATP. And you're going to need to include all of that when you do your ATP math. So odd chain fatty acid always results in just 1 propenyl-CoA. The propenyl-CoA, in order to not waste those electrons, those carbon-hydrogen bonds will be made into succinyl-CoA, costing an ATP. And then that succinyl-CoA will enter into the TCA cycle sideways, not at the beginning, but where succinyl-CoA is, and you'll get a spin around up to the top, not all the way around, but only up to the top. So you get a GTP, an FADH2, and an NADH. And then we meet one more vitamin. So in order to get from propenyl-CoA to succinyl-CoA, we meet vitamin B12. So vitamin B12 is also known as cobalamin. because in the structure of it, yikes, it's huge, right? You see that there's a cobalt sitting right in the middle of a heme. Do you see that? Or I'm sorry, not the heme, a porphyrin ring. See the porphyrin ring? It's in blue. The porphyrin, remember it was a porphyrin plus the iron equals the heme. In this case, you get a porphyrin plus a cobalt, gets you a corin. Cool, huh? I think it's cool. The same with all of our other vitamins that turn into coenzymes. Cobalamin here, coenzyme, they are always attached to nucleotides. So NADH is nicotinamide adenine dinucleotide. FADH2 is I just lost my flavin adenine dinucleotide. So this coenzyme that includes vitamin B12 has 2 nucleotides as well. And the chemistry for what vitamin B12 can be used for in an enzyme, because it's a coenzyme, so it's a coenzyme because there are carbon-hydrogen bonds. It's not amino acid. It helps the enzyme function. The chemistry of it is actually really interesting. So what you're seeing is just in general with a vitamin B12, you get a swap of the placement. So here, H and X could be any functional group, are switched to X and then H, and you see the same thing happening with this methylmalonyl-CoA mutase to get to succinyl-CoA. The places are just swapped. Anyways, vitamin B12 needed for odd chain fatty acid oxidation. And here's a slide that again shows that for odd chains, right here, they go through beta-oxidation just the same as an even chain would, each round spitting out 1 acetyl-CoA until the final round, which gives you 1 acetyl-CoA and 1 propenyl-CoA, whereas the even numbers only ever spit out acetyl-CoAs. Okay, the last two fatty acid nuances are long chains and branched, and I'm going to put them together because both long chain and branched are broken down in the peroxisome. I should actually say broken down first in the peroxisome. And the reason for that is it makes them shorter chains, and it spits out a little bit of acetyl-CoA that can go to the mitochondria for complete oxidation. Okay, so here's an example of a long chain or a very long chain. These have to be a minimum of 22 carbons long. Okay, so if the fatty acid that you eat or that you've made has more than 22 carbons, it first has to go to the peroxisome, gets chopped down into smaller pieces, and those smaller pieces then go into the mitochondria. Phytantic acid right here is an example of a branched chain fatty acid. You can see the methyl groups coming off of it. Branched chain fatty acids tend to come from plants. Long chain fatty acids tend to come from dairy, meat, fish fat, things like that. What I just want to point out with a branched fatty acid is that you'll notice if I do my rounds where I cut after the 2 carbons on the main chain, what happens during the branch is that I get a 3-carbon intermediate, or a 3-carbon-CoA, and that is going to be propenyl-CoA. So sometimes in these in-between one, like that one will lead to an acetyl-CoA. But where the branch points occur, at least in phytanic acid, you would get a propenyl-CoA because there's 3 carbons there, and you're still going to throw a CoA on it. Okay, so let's talk about the peroxisome a little bit because it's kind of cool. All right, so here's a really interesting diagram where you have the mitochondria on the left and the peroxisome on the right, and we're going to do some similarities and differences, and then I'll walk you through this. So similarities and differences between the peroxisome and the mitochondria. So some similarities are they both do beta-oxidation. And they both have 4 steps for this. Remember, oxidation, oxidation, hydration, oxidation, dialysis. So they both have all 4 steps. They both yield acetyl-CoA. So you can see, here it is. They both yield acetyl-CoA and NADH. So the second oxidation step, they both yield NADH. The difference is the peroxisome does not yield FADH2. Instead, the electrons are passed to hydrogen peroxide, H2O2, which is immediately broken down into water and oxygen using an enzyme called catalase. You should know this enzyme. So you'll notice on the very first step, again, here's the mitochondria, and here's the peroxisome. The peroxisome is another membrane-bound organelle that's inside the cell. In beta-oxidation in the mitochondria, you make FADH2. In the peroxisome, the electrons are passed to FADH2, and then they're passed to peroxide and to make a peroxide. And peroxides are reactive oxygen species, and they cannot last for any great amount of time because they cause a lot of damage in the cell. So catalase breaks it down into water and oxygen, 2 very harmless molecules. And the other difference is that the peroxisome it lacks an electron transport chain and an ATP synthase. I'm running out of space here. So there's no ATP made in the peroxisome. Okay, so let's look at this again. So we haven't gotten to electron transport chain or ATP synthase, but we will. That's coming up next. But you'll notice that the electrons from NADH in the mitochondria are passed to the electron transport chain. That's this part. And then that is used to power ATP synthase to make ATP. Same thing with FADH2. Electrons are passed to an electron transport chain. That's the oxygen to water. And then that is used to power ATP synthase. And that is happening in the mitochondria. You'll notice that nothing happens over here in the peroxisome. So once the peroxisome has made acetyl-CoA, it gets transported into the mitochondrial matrix to go to the citric acid cycle, and then the citric acid cycle also feeds the electron transport chain. All right, so that leads us to our second disease. So our second disease is disease associated with beta-oxidation in the peroxisome, X-linked adrenoleukodystrophy, XALD, or just ALD. There are actually a few different kinds of ALD or XALD, and we're going to focus mostly on the child cerebral form of this. There's child, there's adolescent, and adult, and there's like a few different phenotypes of this, but we're going to focus on this one. Okay, so what this one particularly presents as is a mutation. I'm going to turn the page here. Come back here. All right, so what this presents as is a mutation in the transport protein that allows or should allow very long chain, very long chain fatty acids into the peroxisome. Okay, so what you're seeing in this diagram is there's a cell that's in pink. Here's your mitochondria here. Here's a peroxisome here. Long chain, very long chain fatty acid enters into the cell, becomes a very long chain fatty acid, and then with this mutation is not allowed into the peroxisome. So what that means is that these long chain fatty acids and very long chain fatty acids cannot be broken down. They can't be broken down in the peroxisome, and you can't be broken down into the mitochondria. So what ends up happening in this particular disease is that the myelin, I did not spell that right. Hold on. Let me try again. Myelin, the fat from myelin in the central nervous system is broken down instead. And this has some really devastating consequences. This affects boys. Again, that's the X-linked is the X chromosome. And it results in loss of vision, and then learning and behavioral problems. Let's see. And typically leads to death before the age of 10, or usually by the age of 10. By age 10, again, and this is in the infant version, not the adolescent or the adult version, which those can also lead to death, but typically lead to behavioral disturbances and learning disabilities. Okay, so how do you detect this disease? It's actually kind of tricky to detect it because there are many different phenotypes. But the first thing that is looked for is you check the blood for the very long chain fatty acids. This is also a disease that's being screened for in newborn screening. It's not typically as successful as the MCAD newborn screening because there are different phenotypes related to it. Okay, so there's a few different treatment methods. One is dietary. So again, basically you just restrict fat intake. Don't eat the very long, let's say low fat. But this doesn't cover all the problems. So your body will actually make the fat as well. So not only do you eat some of these fats, but your body makes it. So there's this Lorenzo's oil. It's a movie. I actually haven't seen it. Lorenzo's oil that is an inhibitor of the enzyme that synthesizes the very long chain fatty acids. So those two in combination might be able to help. The most successful treatment actually so far has been stem cell. And this is also a disease that's been targeted for gene therapy because the risk is deemed sort of worth it, like ethically. So because it affects babies and children, and the life expectancy is very short, and the symptoms are really awful, it's been one of those diseases that they've sort of deemed, well, we might as well try it because the risk is worth it. But so far, the stem cells has been the most successful. It's not great, but it's been the most successful of the potential treatments. Okay. All right, so switching gears just a little bit into ketone bodies. This is the very last thing. And so ketone bodies are, I don't know, let's call them like a byproduct of fatty acid oxidation. And they're typically made during fasting or starvation, particularly from low-carb diets. So low-carb diet, you're not eating any sugar, you've depleted your glycogen storage, and so you're using your fat storage and the fats that you eat for metabolism and to make ATP. And the tricky part becomes when your brain needs sugar. So your brain has an obligate relationship to glucose. And that's where some of the acetyl-CoA that comes from beta-oxidation is broken down into what are called ketone bodies. And the 3 ketone bodies that you'll need to be able to recognize the structures of are acetoacetate, acetone, and beta-hydroxybutyrate. And they're called ketone bodies because they have ketones in them. This can become a ketone. And these ketone bodies are particularly blood soluble or water soluble, unlike the fat. So they readily travel through the bloodstream to get to the brain. The heart will also use ketone bodies for energy if it needs it. And they cross the blood-brain barrier readily. They're very small. Acetone is actually exhaled or excreted. So it's mostly beta-hydroxybutyrate and acetoacetate that are the 2 that are going to be used by the brain for energy. So I'm just going to kind of say that all again. So ketone bodies are a byproduct of fat metabolism. And this happens when you're on a low-carb diet or you fast or you're starving because you run out of glycogen and your brain needs sugar, but you're not eating sugar. And so it will take the fat reserves in your body or any fat that you eat, and it will convert some of acetyl-CoA into these 3 ketone bodies. And these 3 ketone bodies are water soluble. They travel through the bloodstream. They cross the blood-brain barrier to get to your brain, and I'll show you how you can get energy back out from them. Okay, so let's see. Let's focus in on this one. So here's fatty acids. Oh, this is really pixelated. Sorry about that. So fatty acids that you've eaten or that come from your adipose tissues go to the liver. Some will be broken down, beta-oxidation, go to TCA cycle. I should be using a different color. TCA cycle, be made into ATP. However, if you're starving, some of the fatty acid will be siphoned off to undergo ketogenesis to make ketone bodies. And some of those ketone bodies will go to the heart, and a good chunk of them will go to the brain. And this is because your brain has an obligate relationship with glucose. If you're not eating glucose and you can't make it, then a lot of the energy is going to have to come from these ketone bodies in order to keep your brain functioning. And this diagram right here is kind of interesting. So ketone bodies can help supply up to 60% of the ATP. So what we've normally gone through is glucose goes in, glycolysis, fermentation, and then we have the PDC. It goes to the TCA cycle, electron transport chain, ATP. We're going to talk about this next time. Ketone bodies in only 3 steps can be made into acetyl-CoA. So this is, just so you know, this is the PDC. This is the bridge step. Can be made from 3 steps back to acetyl-CoA, go into the TCA cycle. So you can get energy ATP out of a ketone body. But because your brain has this obligate relationship, I have no idea what I just clicked on. Yikes. Is this still going? Oh dear. But because your brain has an obligate relationship with glucose, you still need to be making sugar. And eventually we will also talk about how you can make a little bit of how your body can make its own sugar, but it can only make just a little bit of it. Okay, so this leads to ketoacidosis. So what happens if you go really long time without being able to break down sugar, which is what diabetes is, or you just don't eat sugar and you're only relying on lipids for ATP synthesis. Okay, so ketoacidosis, again, this happens during fasting or starvation, or a super low carb diet. And this also occurs for people with untreated diabetes. Which I'm just going to abbreviate as people with untreated diabetes. They have sugar in their bloodstream, but the sugar or the glucose doesn't enter the cell. So essentially, they're starving for sugar while being immersed in sugar. So the brain cannot get the glucose into the cell for glycolysis and the TCA cycle. So ketoacidosis happens when starving, fasting, low-carb diet, untreated diabetes. Again, the acetyl-CoA is made into these three ketone bodies. Acetone, like I said before, is exhaled or excreted. And you may have heard, like, people with diabetes, they have really sweet-smelling breath, and that's actually because of the acetone, or partially due to the acetone. If you've ever gone on like a super low carb diet, you'll notice that like your breath kind of has like a different, it just feels a little bit different, and that's because you're exhaling acetone. And you probably worked with acetone in the laboratory before. If you spray it on a benchtop, it evaporates like almost right away. Okay, so these are, so acetoacetic acid and beta-hydroxybutyric acid are the two that are left in your bloodstream. And if you have too high of a concentration of these acids, particularly of this, so in your blood, if you have too high of this, your blood pH drops because it's an acid. It's no good. This is what leads to, wow, I just can't spell anything. Acidosis. Okay, and then a high concentration of both of these, particularly the acetoacetic acid, is called ketosis. So you put them together and you get ketoacidosis. And having a really low pH in your bloodstream is a really bad thing. You don't really want to change the pH of your blood because your proteins fall apart and they don't function. And then there's just a huge amount of things that can come from that as negative consequences. Okay, so hopefully this didn't feel too discombobulated, but we've gotten through all of fatty acid oxidation. We've gotten through some fatty acid nuances, some diseases, and we've also gotten to ketone bodies, which is something that I sometimes are not able to get to. So, yep, okay, sounds good. I'll see you later. Bye-bye.